Okay, so this should look familiar. This was glycolysis. So at the end of glycolysis, now if oxygen is available, instead of doing fermentation and completing an anaerobic respiration pathway without oxygen, we are going to go into a pathway where oxygen is present. And then if that's the case, step 2 is called the Krebs cycle. Important to note, if we look over here, so from glycolysis, we get these pyruvate molecules. Since glucose is broken into 2 pyruvate molecules, so we started with glucose, we ended with 2 pyruvate molecules, we're always going to have 2 turns of the Krebs cycle. So I'm going to have you guys learn numbers of compounds when we look at the cycle, and so that's going to be where all of our numbers are doubled if that doesn't make sense. So on your test, I'm going to ask you guys to know all of the things in the boxes. And then these 4 boxes also. My in-person students have spent a lot of time memorizing these steps. I've given them a lot of tips and tricks on memorizing things. If you guys have any questions for maybe ideas with that to maybe improve your study skills, feel free to send me a message. I can tell you guys kind of what we were doing. But as far as doing that on a instructional video, I feel like that would be hard to do. So anyways, we start with pyruvate. It's converted into a different compound, a 2-carbon compound called acetyl coenzyme A. In this step, we used NAD+ to give us NADH, and we lost a carbon, so CO2 was produced. This is technically not Krebs cycle. The Krebs cycle, if you look down further, is the obvious cycle portion. So the first step of the Krebs cycle, acetyl coenzyme A is combined with a 4-carbon compound called oxaloacetate to give us a 6-carbon compound called citrate. So 4 carbons plus 2 carbons gives us 6 carbons. 6-carbon citrates turn into 6-carbon cisaconate, which is turned into 6-carbon isocitrate. In between these steps, we have an NAD+ molecule being used to give us NADH. And we have CO2 produced because we're going down in carbons to 5-carbon alpha-ketoglutarate. Same thing here. We're going to create NADH, and we're going to create CO2 before we get to 4-carbon succinyl coenzyme A. In the next step, we're going to combine ADP and a phosphate group to give us ATP. So that's a big thing. I'll definitely have questions about that. So then succinyl coenzyme A is turned into 4-carbon succinate. In between this step, a molecule called FAD, which is a lot like NAD, is converted into FADH2, and that's going to matter. That's going to be something I'm going to ask you guys about. And then succinate is turned into fumarate, which is turned into malate. We have one more NADH created, and then we're back completing our cycle with oxaloacetate, ready to do the Krebs cycle again. As far as numbers of compounds, so this is going to be something that I'll ask you guys questions on the quiz. Gains of the Krebs cycle. So NADH, if you count them up, remember this one doesn't count. 1, 2, 3. But I'm going to write a 6. And the reason for that is, like we said up here, everything's doubled. So we had 2 pyruvates from our 1 glucose molecule, so you're never just going to have 1 Krebs cycle. You're always going to have 2. So those numbers are doubled. So that's why that's 6 when you can only count 3. CO2s, we produced 1 right here and 1 right there. So there's 2 of them. Remember, that one doesn't count. There's 2 of them, so we're going to write 4 because everything's doubled. ATP, it's just this 1. So I'm going to write a 2. Everything's doubled. And FADH2, there's just the 1, so I'm going to write 2. So make sure you guys know those numbers with these compounds, as well as the name of each compound of the Krebs cycle. My phone, I was going to do this in one video. My phone's going low battery. So before we get into step 3, which is going to be the electron transport chain, I'm going to charge my phone. So there will be a third video. So check that one out after this one.